Okay, so today I will discuss about the methods, different methods of feed protein evaluation in livestock and poultry. That means in non-ruminant and ruminant as well. So in this chapter, I will take almost 3 class. So this is the part 1 of this chapter. So first of all, how we can evaluate the protein quality in monogastric animals, and what are those methods for evaluation of protein quality in non-ruminant. And after finishing the non-ruminant part, we will discuss about the ruminant part. So how we can evaluate the quality of the protein in ruminant as well. So today we will discuss about the monogastric species, how we can evaluate the protein quality. As we all are aware about that, what is the importance of protein in livestock feeding? And protein is one of the major component and main nutrient of the diet. And like energy, protein is one of the major nutrients and very costly. If we think about, if we calculate the cost of the protein in the diet, so cost of the protein is maximum in whole ingredient, and protein is essentially required for maintenance of the body, and for building of the body tissues, the protein is essentially required. For normal maintenance of and for normal metabolism, it is also essentially required. So we can say that protein is very much vital for the feeding of the livestock. And diet that contains different amino acids. And there are mainly 2 types of amino acid, essential and non-essential amino acids, which is present in the diet. But the livestock desires some more quantity of amino acid, which is not available in the ingredients or not available in the source of the diet. So we should supplement those amino acid from the outer side. So that amino acid which is not either synthesized by the animals or not that much available in the source of the diet. So that amino acid is known as essential amino acid, which is supplemented through the diet from outer source. So these amino acids are very much essential for different function of the body, and the protein quality evaluation, which is also depends on the digestible protein quantity. How much protein is digested? Whatever the feed offered to the animal, then how much protein is digested by the system and how much digested protein is being absorbed by that particular species of non-ruminant. So this is very much important. Digestibility of the protein is decide the requirement of the protein, you can say. So because the efficiency of different kind of the protein of digestion and absorption is different, so this is also one of the factor which affect the protein quality and evaluation of the protein in monogastric species. That digestibility of protein varies with the type of the protein, type of the diet, and source of the protein. So in order to this, we can evaluate the protein quality by protein efficiency ratio. We can calculate by this ratio, and net protein retention, gross protein value, and several other different methods we will discuss. So by that way, we can evaluate the protein quality in monogastric animal. So what is the protein efficiency ratio? How we can calculate it? So protein efficiency ratio is equal to the gain in body weight, gain in grams, unit is grams. So gain in body weight divided by protein consumed in grams. So this is protein efficiency ratio. What is the net net protein retention? So net protein retention is equal to weight gain of test protein group. Test protein group diet minus weight loss of non-protein supplemented group divided by weight of protein consumed. So by that way, we can calculate net protein ratio, net protein retention, where TPG is the test protein group and NPG is protein free diet group. So by that way, we can calculate PER and NPR. Next to this is gross protein value. How we can calculate gross protein value? Actually, gross protein value is equal to A divided by A naught. So where A is grams increased weight gain divided by gram test protein, and A naught is gram increased weight gain divided by gram casein. Casein is the purified diet. It is the source of the protein, pure form of the protein. This is the casein. So how, by that way, we can calculate the gross protein value. And you can see here the example how we can calculate it, the GPV, that live weight gain of the birds, which received the basal diet. Basal diet means the normal diet, which is required for maintenance of the bird. So basal diet which contains 80 gram crude protein per kg, and that to be compared with the diet, the bird receiving the basal diet plus 30 gram per kg of the test protein. That basal diet plus 30 gram per kg of the test protein, and other group which is receiving the basal diet plus 30 gram per kg of purified diet, purified protein, that is casein. So between two we can compare it, and by that way we can calculate the gross protein value. So how much the extra live weight gain per unit of supplementing the test protein, and extra live weight gain per unit of the supplementing purified diet, that is casein, and we can calculate the GPV of the test protein by differences. So like that we can calculate the gross protein value of particular diet in watts. And now come to the nitrogen balance. What is the importance of the nitrogen balance in monogastric species, and how we can calculate it? Actually, which is consumed in the food, which is measured by that the amount of nitrogen consumed minus amount of nitrogen provided through feces, urine, and even either milk. In milking animal, we can egg, even in case if we talk about the poultry, then different sources of the nitrogen, either egg, like that. So amount of nitrogen consumed minus amount of nitrogen provided through different sources. So by that way, we can calculate the nitrogen balance in the body. So one term is nitrogen equilibrium. So nitrogen intake minus if that nitrogen intake which is equal to the nitrogen output. So by that way, in this, we can say that this is the animal is in nitrogen equilibrium. So there is no traces of the nitrogen losses from the body. Whatever the intake and whatever the output, but no losses from the body. So balance of nitrogen takes place in the body of the animal. So in such case, we can say this is the animal is in nitrogen equilibrium. And once the nitrogen intake and nitrogen outgo is less from the intake of the nitrogen, then we can say that the animal is in positive nitrogen balance. And when nitrogen intake and more amount of the nitrogen excreted from the body, then animal is in negative nitrogen balance. So we can say that animal either in positive nitrogen balance, equilibrium, in equilibrium condition or negative nitrogen balance. So health of the animal will also depends on the presence of the nitrogen or presence of the protein in the body of the animals. So there are different factors which affecting the digestion, which affecting the balance trial while calculating the different parameters. So there is the if inadequate adaptation trial period of the animal. So we should take into account that the adaptation period should be whatever the minimum required for any trial, and what is the environmental condition while running the trial for calculating or evaluating the protein quality in particular species. Collection of the samples, collection of the feces, urine, etc. And proper weighing should be done. Otherwise, these are the factors which affect the evaluation of the protein. And a storage of collected samples, how we can store it. So we should adopt the standard operating procedure, whatever required for collection, storage, sampling. So SOP should be there, and we should follow the SOP while evaluating the protein quality of the diet in the non-ruminant species. So very much important, that is the biological value is, by biological value we can also estimate it, what is biological value. So the amount of protein which is consumed by, or we can offer to the animal, that is being utilized by the animal body for synthesizing of different body tissues. So how much amount of protein which is absorbed by the system for the synthesis of the body tissues. So this is the absorbed quantity of the nitrogen which is retained by the body. Simply we can say that the offered protein which is absorbed by the or retained by the body is biological value. So biological value, we can calculate the biological value. Before calculating it, we should go for the balanced trial. So feeding trial should be essential, and balanced trial, we should perform the balanced trial while calculating the biological value. So biological value is equal to nitrogen intake minus fecal nitrogen minus metabolic fecal nitrogen minus urinary nitrogen minus endogenous urinary nitrogen divided by nitrogen intake minus fecal nitrogen minus metabolic fecal nitrogen. So by this formula, we can calculate the biological value of a species or of a particular diet in particular species. So we should you should remember this biological value formula, and this is very important biological value, how we can calculate it, and what is the formula of the biological value, and what is the biological value. Definition may also ask in the examination. So you must remember this biological value, and what is the MFN and endogenous urinary nitrogen. We will discuss later on in detail, and what should be the value of that EUN and FMN in particular species. Here you can see that how we can calculate the biological value for maintenance and growth in. This is the example in rat. So like that we can calculate in other species also. So how much food consumed per day, how much nitrogen present in the food, and daily nitrogen intake, total nitrogen excreted in urine, endogenous nitrogen excreted in urine, total nitrogen excreted in feces, and metabolic fecal nitrogen excreted through feces. So by that way, we can calculate the biological value. So these data should be required and must be required for calculating the biological value. You should remember some points that the biological value of any food protein, which depends on the availability and number of the amino acid, what kind of the amino acid present in the diet. So that availability and uh, type of the amino acid which affect the biological value. Like closure the amino acid composition of fruit protein that approaches the body protein. So the higher will be its biological value. And the product of biological value and digestibility in terms of net protein utilization, and the proportion which is for nitrogen intake, which is retained by the animal, that also important for calculating the biological value. Here you can see that the biological value of the protein, examples of biological value of the protein of various food, like milk in case of the growing pig. Like milk, the biological value is 0.95 to 0.97. Or we can say that if you can calculate in percentage, then it is 95 to 97%. Like fish meal, soybean meal, cotton seed meal, linseed meal, maize, barley, pea. So this is the example of the biological value, how it can be varied from the diet to diet and ingredient to ingredients. Next to biological value, that is the chemical score, and how we can calculate the chemical score. That chemical score, which is considered that the quality of the protein, which is decided by the amino acid, which is present in particular diet. So we can say that the greatest the deficiency when compared with the standard. And a standard generally used as the egg protein, which is considered as a standard protein. But many workers and many researchers now they use the standardized amino acid mixture. So from the egg protein to coming to the amino acid mixture as a standard. And the content of each of the essential amino acids, which is as the protein which is expressed as the proportion, that is in the standard, that the standard pattern ratio, and the lowest proportion which is taken as the a score. So you should you just go through this line. The content of each of the essential amino acid of a protein is expressed as a proportion of that in the standard. That is a standard pattern ratio, and the lowest proportion taken as the score. So very much important. And how we can calculate the chemical score here, you can see that the examples of calculating the chemical score in wheat protein. So in wheat protein, that the example of essential amino acid, which is greatest deficit, is lysine. So wheat protein is deficit in lysine. That is, lysine is essential amino acids. And the content of lysine in egg and wheat protein are 72 gram per kg and 27 gram per kg. In case of wheat protein, 27 gram per kg. In case of egg, 72 gram per kg. And the chemical score for wheat protein is, then we can calculate the chemical score of wheat protein, that is 27 divided by 72. So the chemical score coming to 0.37. So like that, we can calculate other protein sources also, the chemical score of other protein sources. And we should take the egg protein as a standard. And like that, the useful for grouping the protein, but while grouping the protein, suffer the serious disadvantages. That is, the no account is taken of the deficiencies of amino acid other than that is greatest deficit. So this is also very much important. So we should kept in mind while calculating the chemical score of diet. After chemical score, coming to the essential amino acid index. What is that? Actually, essential amino acid index it is the geometric mean of a standard diet, like egg, a standard pattern, and the ratio of essential amino acids. So it is geometric mean. And it has advantage of predicting that the effect of supplementation in combination of protein, and it has disadvantage also that the protein of different nature or very different amino acid composition which may have the same or very similar index. So this is the index, essential amino acid index. Name itself says this is the index. And it has disadvantage that very different amino acid composition which may have the same or very similar index. And chemical score and essential amino acid index, which are based upon the composition of the gross amino acid. So composition of the amino acid is very much important for calculating either chemical score and essential amino acid index. And composition of amino acid varies with the diet. Different diet or different ingredients have different composition of amino acid. So these parameters also vary with the nature of the diet. So this is about the today class. And if you have any question, then you can ask me, or either we can stop the sharing.